All right, so we are starting the muscles and muscle tissue chapter. This is chapter 9. We will get about 30 slides into this, and then we'll pick up with neuromuscular junction on Thursday. So we are going to be focusing mostly on the skeletal muscles section, but also keeping in mind that there are two other types of muscle, cardiac and smooth. So with the skeletal muscle, this is what's attached to bone. This is what we talk about being your the muscle that you can voluntarily move. Whereas cardiac and smooth muscle are involuntary muscles. Your autonomic nervous system controls those things. So when we look at the microscope pictures, you can see that all these different muscles look different under the microscope. So with skeletal muscle, it's a long cylindrical multinucleated cell. So you can see each one of these cells. There's 5 cells here, and each one has multiple nuclei. That's what these purple circles are. The striations are these stripes that are going more vertically compared to the horizontal cell. With cardiac muscle, the muscle in the walls of your heart, these muscle cells are branched. So you can see here is a cell, and it has more than one nuclei in it, and it also is striated. So the multinucleated striated portion is very similar to the skeletal. And then smooth muscle, like the muscle in your blood vessels and the hollow visceral organs, aside from the heart, this is where we see the smooth muscle. And it has this fusiform shape where it tapers at the ends. It only has one nucleus per cell, and there are no striations. So those are the big differences between looking at these different cells. All right, so focusing back in on the skeletal muscles. So skeletal muscle cells are also known as muscle fibers. So don't get confused when you hear the word muscle fiber, just think of it as a muscle cell. And you'll see these portions of words, sarc, myo, mice. These are all parts of words that indicate we're talking about skeletal muscle. So some characteristics of muscle tissue, it is excitable or irritable, meaning it can receive stimuli and then respond to it. So think of touching a hot stove. Your body says, okay, there's danger, and you use your muscles to get yourself out of the danger and pull your hand away. Muscles are contractile, so they can shorten forcibly in order to move. And then they're also extensible, meaning they can be stretched. And then the elasticity portion of this means that they're able to recoil and resume their original resting length. Skeletal muscle has a few different functions. It's responsible for allowing us to move, maintaining our posture, stabilizing your joints. I was just talking to somebody about this yesterday, in that muscle strength is crucial for decreasing stress into your joints. And then also generating heat to keep your body at 98.6 degrees. Muscles also assist in the movement of blood and lymph fluid back to your heart. So especially important in your legs, where your muscles have to pump that fluid against gravity. So in your lower legs, we have these veins that contain valves. And what will happen is the blood will hang out here. You'll have a muscle contraction, and it'll be pushed, or the fluid, sorry, the fluid will be pushed into this next chamber. Okay, we get another muscle contraction, and it's pushed again into this next chamber until it can leave the area. So the muscle contraction helps both venous blood move as well as the fluid move up. We can use different devices, like there are, these leg pumps. For athletes, there are ones called Normatec that have this chambered compression that starts at your feet and then slowly works its way up your legs. But there are also like medical devices that are used in hospitals to decrease the incidence of blood clots. And then, of course, you have your support hose, these TED hose, that also help push the fluid back into your vessels and help it get up back into your heart. So you might notice, especially if you're sitting for long periods of time, that you might have a fluid collection at your ankles. It's simply because your muscles are not working. So they say, okay, if you're on an airplane for a long time, pump your ankles, pump your legs, get up and walk so that you don't have that and you don't increase your risk of blood clot. Muscles are also protection for your abdominal viscera. So you've got this bony cage here in your ribs, you've got a bony cage around your pelvis, but there's no bony cage around your belly. So we've got several layers of muscle that act to protect all of that stuff that's in there. So skeletal muscle, like I said, is also known as a muscle fiber. So we have these bundles within the muscle to give us a large muscle. So we have endomysium, which surrounds the actual muscle cell. That's like this covering to hold it together. Our next layer is a fascicle, and the fascicles are a collection of muscle fibers. So you can see here it's like a container of pipe cleaners, I feel like. And that container is the perimysium, and it's holding all of these muscle fibers together. Collectively, it's known as a fascicle. And then we get our big muscle here, which are bundles of fascicles. And we have epimysium that covers the outside of your actual muscle, and it collects at the end to become the tendon. The tendon is what attaches the muscle to the bone. So I'd like you to pause this video and label using these terms: muscle fiber, fascicle, and muscle. And then also label endomysium, perimysium, and epimysium. So take a few minutes, pause the video, and label that. That is really important that you understand that organization. All right, so hopefully you had an opportunity to do that. So I want you to think of steak. You guys are never going to look at food again. Think of this as skeletal muscle. This tissue around the steak is the epimysium and the fat. The whitish marbling within the steak, that is perimysium and also fat. And then when we're looking at this, you cannot see the endomysium because it's so small. All right, so most skeletal muscles span the joints. So when you need to move a joint, obviously like your bicep here, it has to go further than just your humerus here. It has to actually go into your forearm. So it can move this like a lever. So we will see attachments of the muscles to the bone in two places. One place is the origin, and that's the immovable end, so kind of the anchor point. And then we also have our insertion point, which will be the movable end. So a contraction should bring the insertion closer to the origin. So if you were to flex your arm, these muscle fibers contract in this direction, and this insertion will get closer to the origin. All right, so that is how muscle directly attaches to bone. Muscle also is indirectly attached, and that's when we see this epimysium not turning into a tendon, but it extends beyond the muscle and attaches to another muscle. And this is known as an aponeurosis. And we've got one right in the middle of our abdomen called the linea alba that connects our six-pack muscles here, the rectus abdominis, together. If you were to be pregnant, you might notice on pregnant bellies that there's this browning line right here. This is called a linea nigra. And this is when estrogen activates your pigments to give you this brown line. You can also get it on your face and other areas of your body called melasma. But you get this connection right here over where the linea alba is. So it is called linea nigra during pregnancy, and it does go away after a couple of months after having the baby. Okay, So then you have a more direct attachment of the epimysium being fused to the bone's periosteum, which is how we see the facial muscles work. If you think of like old westerns or things like that, where they talk about getting scalped by the Indians, or to be more PC, the Native Americans, it's because these muscles of your scalp are very loosely connected to the periosteum. So once you have a cut in the forehead or even back here, it kind of just flips up. All right, so muscle can also be directly fused to bone using the periosteum, kind of similar to a tendon. So each muscle in your body is served by one artery, one nerve, and one or more veins. And you can see here on this bodybuilder, we get this veining because these muscles require more blood supply. So you're also going to need more blood return. Also, just because of dehydration and different things that bodybuilders will do, you can make these veins pop a little more and seem more obvious. All right, so let's take a look at that muscle fiber. So remember, a muscle fiber is a long cylindrical cell. So this cell, this thing, is a whole muscle fiber. It contains these different organelles. And you'll notice some of these are very similar to any other cell, but we have some different names. So the sarcolemma is like the cell wall. It's underneath the endomysium. And then the actual organelles, which are found in the sarcoplasm, which is just another name for cytoplasm. So that sarcoplasm contains multiple nuclei. So you can see here, we've got multiple nuclei in there. The myofibrils, which are proteins, that's what all of these things are in here, and that's about 80% of the fiber volume. We have mitochondria, which are these orange things. Glycosomes, which are sugar storages for our glycogen. And then myoglobin, which gives the muscle its red color, and maybe this picture is trying to represent this as the myoglobin. But this is intracellular storage for oxygen, which is why muscle has that red color. That oxygen makes it red. So think of myoglobin like hemoglobin, but just found in your muscles. So that sarcolemma, like I said, that is like the cell membrane. You can see that here. This forms tunnels throughout that muscle fiber known as transverse tubules or T-tubules. And these are extensions into the muscle fiber. This is important because nerve impulses come to the muscle, but then that nerve impulse spreads down the length of the fiber because of the sarcolemma, and then it dives down into the muscle fiber through these T-tubules. And then the sarcoplasmic reticulum, this is another new one, kind of like endoplasmic reticulum. That's all this blue weaving. And what this SR is it forms a triad with the T-tubules. So you can see these yellow lines. These are the T-tubules coming down. And we have that sarcoplasmic reticulum on either side. And what this SR is temporary storage for calcium. Remember, blood calcium levels win. We need calcium in our blood for muscle contraction. Okay, the myofibrils, these have the ability to contract. That's what all of these protein rods are within the muscle cell. And they're made up of repeating units called sarcomeres. So when we look at this, a sarcomere goes from Z disc to Z disc. So that's one. Here's another one, Z disc to Z disc. That's one. And then we have kind of a partial third here of a sarcomere. These are just the units of contraction within muscle. So the sarcomeres are composed of proteins that can contract, and again, they are the ends, like the bookends, are the Z discs. You can also call it a Z line or a Z band. There are two different types of proteins, the thick filaments and then the thin filaments. Thick filaments are known as myosin, thin are known as actin. And this banding pattern is due to the arrangement of filaments. Remember, that was called the striations, and it's because when these myofibrils are all lined up and stacked on top of each other, these line up perfectly. And you can see that there's a little bit of different coloration in here because of the arrangement of myosin and actin. So when you kind of look far away from this, you have a darker region known as an A band, which spans this bright red color. That's the A band. And then you have the I band, which is a lighter region. So that kind of surrounds the Z disc. It's more of this like peachy orange color. So when we look at one sarcomere, this would be the Z line, okay? This is the A band, and this is also known as the protein myosin. So when we look from here all the way over to here, this is the A band. And then the I band is going to be right next to it. It's going to start here, and it's going to go around that Z line. All right, so remember that thick myosin, the myofilaments, that's what makes up this A band. So what we have here is two interwoven tails that are made out of peptide chains, and then there are heads, and these are also peptide chains. And when we get this clumping of the myosin, it ends up looking like a thick filament, which are several myosin proteins together. And these heads are going to form what are called cross bridges. This is where the myosin is going to reach out and bind to the actin. So if we go back to this slide, the actin is this blue stuff that goes in between the myosin. So the dark band is actually where myosin and actin are overlapping. That's what gives it that dark appearance. Okay, on the heads, aside from binding to actin, it's also a binding site for our old friend ATP, and also the ATPase enzyme to break down ATP. And then the thin myofilaments are composed of actin, and these look like, they almost look like olives, okay, all together. And they go in kind of a helix, so you can see how they are connected together and they almost rotate. These contain the binding site for myosin, which if you think of this as an olive, that binding site is like the pimento right in the middle, here where my cursor is. And that binding site is actually being covered up by this other protein called tropomyosin. That's this orange thing. So when we don't want a muscle contraction, we're going to cover up that myosin binding site so that it cannot bind. And then we also have troponin, which is kind of like a clip. That's this yellow thing. It's holding the tropomyosin in place over that actin, that binding site for the myosin. So the way muscles contract, this is called the sliding filament model or theory of muscle contraction. So once a muscle is stimulated by a nerve impulse, that causes those myosin binding sites to be exposed. So that way the myosin heads can reach out and bind to actin. So each myosin head binds and detaches several times. So it's kind of like a ratchet that generates tension when you're like tightening something down to the roof of your car or whatnot. So it's going to slide the actin filaments every time that head reaches out. It's going to grab the actin and bring it in. So you can see that the actin, that's what this red line is, it's coming in from two different sides to meet the myosin, which is this gray line. So each myosin head, they're going to almost reach out in opposite directions like tug of war, grab the actin, and bring it in towards the center of the sarcomere, which is right here. Okay, so the direction of muscle shortening, you can see here, this is a contracted muscle. These Z lines have gotten closer together because of how the actin is the myosin is bringing the actin in closer. So it's like the actin is sliding next to the myosin. Okay, so here again, you can see a relaxed muscle. Here's a muscle that's contracting, the Z lines are getting closer, and then here's a fully contracted muscle where the Z lines are even closer, and now the actin filaments are actually overlapping in the middle of the muscle. Okay, so this is a nice little video for you. I'm not going to click on it because I think it'll disrupt my recording, but you can watch the way that this sliding filament model works. Here you can see a fully relaxed sarcomere versus a contracted muscle fiber. All right, so a couple of questions for you guys. The muscle fiber is primarily made up of the organelles blank. All right, so next one, what is the general function of the myofibril? And then label this myofibril. Okay, so when you're looking at this, the biggest thing with labeling the myofibril is being able to identify this Z disc. The M line, which I didn't talk about yet, but that's the middle of the sarcomere, M for middle. The A band goes around the M line and it spans the myosin, and then the I band is the light band and it goes around the Z disc. So find a blank myofibril and try to label those things. The H zone actually disappears as well as the I band disappears during a muscle contraction because here the actin would overlap, so you wouldn't see this H zone, which is just the space between the non-touching actin. And then this space around here, because the Z lines come together, this would also disappear because the actin is pushed in so far. All right, so go ahead and pause this recording so that you can go through and answer. The questions are continued on this slide. And we'll go over these on Thursday. Okay, so now that you've answered those questions, hopefully, we're gonna just finish up this lecture. So skeletal muscle contraction, this is called excitation-contraction coupling. And it's when a muscle is excited by a nerve and it leads to a contraction. So both neurons and muscle tissue conduct electrical current. So the neuron conducts electrical current to the sarcolemma. And then from there, the sarcolemma can continue that electrical conduction throughout that muscle cell. So it moves ions across their cellular membrane. So we're going to have this movement of ions to keep that nerve impulse going. Okay, so our goal is to be able to describe step by step how a nerve impulse spreads along the skeletal muscle, resulting in contraction. Okay, so again, I'm just going to start this to introduce the neuromuscular junction, which we're going to start on Thursday. So muscle fibers are stimulated by motor neurons. So if you guys have ever seen The Theory of Everything, it's such a great movie about Stephen Hawking and him having ALS, which is a disease of the motor neurons. And you lose the ability to contract your voluntary muscles, your skeletal muscle, and your smooth muscle or cardiac muscle is unaffected. But eventually that becomes impaired because of how important your skeletal muscles are for those things to be able to work. All right, so a nerve contains thousands of motor neurons. So one single nerve can have several branches within it. So a motor neuron's axons branch as they enter the muscle. So you can see here we have some branching, motor unit 1, motor unit 2, and each one of these motor units is going to be in charge of several muscle fibers. And just depending on how precise those muscles need to move, you'll see less branching and just more motor units dedicated to an area that needs to move better. So each branch forms a neuromuscular junction with a single muscle fiber. So here's a neuromuscular junction, here's one, here's one, here's one, here's one. So those are all considered neuromuscular junctions. So the parts of this neuromuscular junction include the axon, which it becomes the end of the motor neuron. And there are these vesicles inside the ends of the motor neurons that contain the neurotransmitter acetylcholine, ACH. We're going to talk about this guy a lot during this chapter. And then we have the motor end plate of the muscle. So this is the muscular portion of the neuromuscular junction. So the axon comes in and communicates with the motor end plate. This is the specific area on the sarcolemma, which is the cell membrane, that contains the receptors for this neurotransmitter. So this neurotransmitter travels from the nerve to the muscle to start that reaction or that electrical stimulation within the sarcolemma. And it is separated by a space. The axon and the motor end plate are separated by a space called the synaptic cleft. So you can see that here. Here is the end of the motor neuron. Here is the motor end plate. And then here's that synaptic cleft, that space. Okay? And here are those acetylcholine vesicles bursting. Okay, they're going through exocytosis, traveling through the synaptic cleft and finding a receptor on the motor end plate. So this is how we go from an electrical reaction down the nerve to a chemical reaction in the synaptic cleft back to an electrical reaction, which will start out that electrical impulse down the muscle. So if we look step by step here, so this will be the last thing that I go through. So when we look at the step by step, we have something called an action potential. That's that electrical signal going down the motor neuron. And when it gets here, it opens the voltage meaning it needs that electricity, voltage gated calcium channels to allow calcium to enter the end of the axon here. When calcium enters, that tells these vesicles to go through exocytosis and release into the synaptic cleft. It diffuses from an area of high concentration to low concentration and binds to these receptors on the sarcolemma. What this will do, this binding of acetylcholine, allows passage of sodium into the muscle fiber and potassium out of the muscle fiber. Okay, so now we're changing the electrical charge of that cell. So acetylcholine effects are terminated by this enzyme called acetylcholine esterase because we don't want this to continually stimulate the muscle. We only want that, one single muscle contraction. We don't want like tetanus where your muscles are in a constant state of contraction. So these are those steps that I just laid out for you with a video. Please take some time to watch that so it's a little bit more dynamic for you. And then again, this is just that the acetylcholine is destroyed by this enzyme called acetylcholine esterase. And the choline will return back into the axon to be recycled, and this just prevents muscle fibers from being in a constant state of contraction. Okay, so what I'd like you guys to do for Thursday is come to class with those practice questions answered. So that is on slide here. So slide 20, what is this? 26 and slide 27. So 7 practice questions that we will go over first thing, and I'll make sure that you completed them. And don't forget your next case study is also assigned, and this will be due with the next test. So for Thursday, what I'd like you guys to bring is your test that I had you take home with you. Bring your case study, and then bring those practice questions. All right, please email me with questions. I guess we actually made it to the action potential portion. But please email me if you have questions after today. We can also go over them on Thursday when we will hopefully meet. All right, have a great day, guys.